osteoclast chewing up the bone, right? So I think one of the review questions asks you to think about what happened if the balance of those bone cells were out of whack, right? Because we have osteoblasts that are helping to lay down the bone matrix, and then osteoclasts that are eating up the bone matrix. So it's this constant remodeling. So I'm going to ask you, what would happen? What happened if you had the same number of osteoblasts, but all of a sudden, like, way more osteoclasts? What do you think would happen to your bone? Bone degeneration, yeah. You'd start to have those osteoclasts kind of winning the battle and chewing up the bone at a rate that meant decreased bone matrix, right? Okay. So on to adipose tissue. So the primary cell type of an adipose tissue are called adipocytes. They form loose connective tissue. Their function is to store energy as fat. So you can see here as the adipocyte develops, it ends up accumulating these lipid droplets, which are full of triglycerides. And those triglycerides expand to take up the majority of the volume of the cell. And in fact, the cell cytoplasm is displaced, right? Because the lipid is what, hydrophobic or hydrophilic? All oil is hydrophobic, yeah, right? So it's the same thing as when you put oil into water, right? Because the cytoplasm is aqueous, hydrophilic environment. So all the oil droplets converge to form one big mass of triglycerides, and then the cytoplasm is kind of pushed to the edge. And so adipose tissue is regarded as loose connective tissue because the cells take up most of the space. There's relatively little ECM there. And in this slide, it's showing kind of the developmental trajectory of fat cells. So fat cells arise from this cell that's called a fibroblast-like precursor. And I'll talk about this type of stem cell in the stem cell lecture. Has anyone here heard of mesenchymal stem cells before? Yeah, I've heard a few people. So mesenchymal stem cells actually give rise to a lot of different connective tissue types. And so these are an example of a stem cell that can differentiate into different cell types according to what genes are expressed. And so at first, kind of the history of these cells, they look long and spindly like a fibroblast. Remember one of the first slides in the connective tissue section was that long spindly fibroblast? So when cell biologists identified this cell, they went, oh, it's like a fibroblast that can somehow differentiate into a fat cell, huh? And then they realized, oh, it has precursor cell properties. It differentiates into different cell types. And so for a while, they tossed around the name fibroblast-like precursor cell. And now this it's more commonly referred to as a mesenchymal stem cell. But if you had in like a petri dish, mesenchymal stem cell and fibroblast, if you're just looking at them, it'd be hard to tell the difference because the cell morphology, the cell shape is really similar. Okay, right. So again, if I say loose connective tissue, does that mean A, more cells and less ECM, or B, less cells and more ECM? More cells, you got it. Yeah, because that loose or dense connective tissue, that's referring not to the density of cells, but like actually the density of the tissue. And what makes a tissue more dense, it's not going to be like the little water balloon cells, it's going to be those tough, fibrous proteins that can pack in really densely in ECM to confer strength to the tissue. All right, you guys good on connective tissue? We're going to move on to muscle tissue. Right? So now we've gone through epithelial tissue, connective tissue. We have 2 more to go, and these are pretty short, much to my dismay. Like a whole lecture to be on neural tissue, but I'm not that big of a boss yet. So, muscle tissue. Muscle tissue is defined as contractile tissue. Hey, Tim, how did your tabling event go today? What? You feel awkward talking with you don't have? Okay. The Science 2 groundbreaking was this morning, and Tian's the pre-PA club. They had like a tabling event there. So it's very fancy. Congratulations. Okay. Contractile tissue, muscle tissues, contractile tissue compose of cells that are always rich in two proteins. I mean, they have a lot of other proteins, but they are, their hallmark is having a ton of actin and myosin. The actin and myosin work together to create the motive force necessary for contraction. Okay, so some muscle tissue is striated, right? It looks like it has this like striped appearance. Muscle tissue that's striated, that actin and myosin form these kind of repeated functional units called sarcomeres. And don't freak out because I'm going to show you a picture of a sarcomere. It's not on your lecture slide because you don't need to know this level of detail yet. I will give you this level of detail later. I just don't want to start talking about actin and myosin and sarcomeres without giving you some sort of context. Okay, so this is an actual image of a striated muscle. And this image right here, it's busy, but what I've highlighted in blue, what I want you to pay attention to, right? So you can see, yeah, there's repeating, it's over here, kind of looks the same over here, there's these repeating units, right? And at the heart of this unit, you can just look between my arms right here, right? You have these actin filaments, right? In this kind of yellow-green color. And then this gray with the little extensions coming off of it, that's myosin. So myosin has these head domains, well, let's call them feet for now. These feet domains that like to bind and walk along actin filaments. So what happens when a muscle cell is stimulated to contract, those myosins will walk out to the edge, and when they walk out to the edge, that force of them walking pushes the actin filaments to the midline. So it's almost like they're walking, and as they're walking, they're like pushing back, right? So they push the actin filaments behind them together, because the way this is constructed, when these filaments walk out this way, this whole thing goes and that is what causes contraction in muscle cells that contain sarcomeres. And then even in the two muscle cell types I'm going to tell you that don't have sarcomeres, they still rely on the motive force that occurs between myosin and actin, right? Myosin is a specialized motor protein that walks along actin. And so that walking force is used to facilitate contraction no matter what muscle cell type you're in. It just so happens that if you're talking about striated muscle, they form this sweet structure called sarcomere. Okay. So that is at the core of contraction. Actin and myosin filaments interacting with one another. So here are the four types of muscle cells. The first big old one you see here, that's a skeletal muscle fiber. And the stripes in it are representing those sarcomeres, the striations, right? So skeletal muscle fibers are responsible for basically every movement that's under voluntary control, right? I do this, that's skeletal muscle. They have this highly elongated shape because their type of cell is called a syncytium. So a syncytium means basically a huge cell that has many nuclei that are not separated by plasma membrane. So they have this huge structure that has all these nuclei and they share a common cytosol. Okay? And it appears striated, again, because of those repeating units of the sarcomeres. Yeah. Yeah, it's the sarcomere is basically this big old structure. So it's not, there's actually more to it than just actin and myosin. There's some intermediate filaments and other proteins that help confer the structure and help hold the actin and myosin in place. But it's the interaction between actin and myosin that cause the contraction and cause movement. Okay. So the one other type of striated muscle cell is heart muscle cell. And it is given that name because it is only found in heart walls. And unlike skeletal muscle fibers, it is not a syncytium, so each individual heart muscle cell has an individual nuclei. and obviously it's not under voluntary control. We don't have to think about our heart beating for our heart to beat. Okay. Smooth muscle cells. So they're called smooth because they're not striated. So in this case, there aren't sarcomeres, but contraction is still facilitated by actin and myosin. They're just arranged differently. So there are a ton of different functions facilitated by smooth muscle cells. So, right, just a few examples are listed here. So your gut has this peristaltic action to move food along it. That's done by the smooth muscle cells that align the gut epithelium on the outside of the gut epithelium to help contract it. When you get scared or excited and the hairs stick up on your arm, the smooth muscle is responsible for that activity as well. Scared. The final type of muscle cell is interesting for a couple reasons. So it's also smooth, like the smooth muscle cell. It's called a myoepithelial cell because this is a muscle cell that is defined within epithelial layers. And it's different than the other ones in its developmental origin, so where it comes from. So all the other muscle cells, smooth, hard, and skeletal, they derive from mesoderm, right? So there's a reason I told you guys about ectoderm, mesoderm, and endoderm. I come back to it a number of times because that helps give us, more insight and context for the cells that I talked about. All those other muscle cells derive from that early embryonic tissue called mesoderm. Myoepithelial cells, it turns out, comes from ectoderm, so they have a little bit different origin, which is what is meant by and are derived from a different tissue during development. And so they are found in the epithelial layers of gland tissue. So they help glands secrete whatever hormone that they need to secrete, right? So on the next image, it shows some cool electron micrographs of muscle tissue. I'll just go to that right now. And first show the myoepithelial cells that are lining the milk-secreting ducts of mammary glands. So this helps lactating moms actually release important hormones and even milk. So, and here are, right, some sweet tissues of those striated muscle cells. So you can really see those striations quite clearly. Smooth muscle cells surrounding nerve fibers. Yep. Cool pictures. Okay. And so, a lot of times when we think about muscle cells, we just think about, right, skeletal muscle and maybe heart, but you don't really think much about how much most of the tissue in your body needs some type of contractility associated with it. So I've put just kind of a laundry list of some of the tissues that aren't commonly associated with a contractile function that actually really need some sort of contraction to help perform its function, right? Arteries, veins, lymphatic vessels, GI tract, respiratory tract, on and on and on. So contractility is important for most of our tissues, actually. So depending on the tissue, depending on the activity, it's going to require a different type of muscle cell. And we have 4 to choose from. Yeah. Okay. So nervous tissue, right? My favorite. It's the fewest amount of slide. So this is the main component of our central nervous system and peripheral nervous system. And there are 2 cell types, so neurons, right, the most popular. But there's also another type of really important nervous tissue that's composed of glia or neuroglia. So these are different cell types that help lend support to our neurons and help them function. So there is a whole laundry list of different glial cell types. I actually just teach you that, hey, there are glia, and then I'm going to talk about the 2 that produce myelin. There are a lot of different glial cell types in our nervous system. Some have immune roles, some have roles in just homeostasis, like maintaining balance of the nervous system environment. It's really, I think, the field of understanding glial biology is kind of booming right now. There's a lot of really interesting work coming out. Okay, so neurons are defined as electrically excitable cells, right? They function in an electrochemical fashion. Glial cells are non-neuronal cells, so they're not electrically excitable cells. They're different. They provide support for neurons. And there's, like I just said, a bunch of different types of ways that they can support neurons. And then I'm going to talk about how the two glial cells, Schwann cells and oligodendrocytes, help action potentials fire rapidly. Right? Neurons are famous for having these axons, right? The two big substructures that a neuron has, so they have their soma, their cell body, and then they have dendrites, which are dendrite. Very technically, I think, means having a branch-like form. And so it's, right, the dendrites look like tree branches. They can be really elaborate, depending on the neuron. There's a bunch of different types of neurons, too, I should say. I mentioned there are a bunch of different types of glial cells. There are even more different types of neurons. And so the dendrites are where a neuron receives information. So that's where another neuron would connect its axon terminal to the postsynaptic neuron. So dendrites receive the neurotransmission. And once a neuron is stimulated to fire an action potential, what happens is you have the action potential rush up the axon. And at the end of the axon is where the neurotransmitters would be released. Again, the axon terminal, if it's participating in neuron-to-neuron neural transmission, it would be synapsing with the dendrite of another neuron. But of course, neurons can also synapse on muscle cells, and they can synapse on their glandular tissue as well. Okay. So that's how neurons function. It's a lot more complex than that, but apparently in 121, that's as complex as I'm supposed to be. We'll see what I can fit into other lectures in there. So functions of nervous tissue, this list is kind of funny because, right, it's as I told my kids, like the brain is the boss of you. Your nervous system controls everything. It's how you process sensory input, or you receive sensory input and then process the output. How you control your muscles and glands. How you regulate your whole body's homeostasis is at the end of the day, they're regulated through your nervous system. And right, mental activity. All mental activities, all of that is responsible. Your nervous system is responsible for all. I was at so Dr. Lundmark teaches this class in, I think, like the kinesiology department. And she wanted me to serve as a judge in this thing she did called the organ debates. So she had like groups of students get together, and these are non-bio majors, so there's a lot of athletes in the class, and they all like team up, and they'll be like, you know, we're the nervous system, and we're competing against like the GI tract or something. And they're given a scenario, and they have to debate which organ system is more important, right? So like I listen to them debate like who is more important for leaving a burning building, getting out of a burning building. And I'm sitting there and I'm like, well, nervous system should win all of these because you just have to say like, I'm the boss of you, like I'm the boss of you, and then just like drop the mic and leave. And they didn't do that. I was like, what are you doing, man? Just tell them you're the boss of them. Anyway, okay. They also got a number of other things wrong, and I was like, can I throw in like a towel? Like, can I? Okay, I'm not gonna talk crap about. Okay, I'm stop. So, they just clearly needed a little bit more molecular biology background. Okay, I digress. Okay. So nerve bundle. So what is a nerve, right? A nerve is actually an enclosed cable-like bundle of axons. That's what a nerve is within the peripheral nervous system. And so it's surrounded by a layer of connective tissue. So here's kind of a cartoony image of a nerve, all the axons running along it. This is an actual image of a nerve, and so they false colored the axon blue So everywhere you see blue, right, it's a bundle of axons, and then each are surrounded by connective tissue. There's blood vessels commingled in there, right? So all this kind of fluffy stuff is the connective tissue. And the white stuff, who can tell me what these coils of white stuff surrounding the axons are? Myelin, yeah, you got it. And so here's a cool image, I think, of myelin. It looks like the rings of a tree. So myelin is like a membranous tissue that's produced by either oligodendrocytes if you're in the central nervous system, or Schwann cells if you're in the peripheral nervous system. And so these cells, their whole job really is to produce these membranes that wrap around and around and around and around the axon. And they form this important, insulating role, right? Because the whole way an action potential works is you have ions, when it's triggered to fire, you have ions that rush into the membrane, right? And get pushed down to the end of the membrane. But, right, if the membrane's semi-permeable, you don't want those ions to just diffuse out of the axon. You need them to come in and rush down. And so what the myelin does, that thick membrane wrapped around and around, it just prevents any ions from moving back and forth across the membrane. So it helps control and regulate the conductivity of that action potential. And so, so here's the most generic image of a neuron, right, that you all are seeing. Here we have the cell body in a dendrite. The way it works is you have, signal comes in, and it's multiple signals coming in. There's a resting potential of a neuron, and you have to get to a certain potential in order for the action potential to fire. If it fires, what happens is you have ions rush into the axon, and you need them to shoot down here, right? And so in order for that to go quickly, you have these cells that wrap around the axon insulated so that they rush, some more can come in, rush, some more can come in, rush, and so on and so forth. And so this is really important when you have long axons. If you have a little itty bitty tiny axon, you might not even need the insulation. So you've heard the terms white matter and gray matter in the brain. You know what the difference is? Myelination. So the white matter, the white of that comes from the myelin. So gray matter means you have a bunch of teeny tiny axons. They don't need myelin in order for the action potential to fire efficiently. When you have big axons, you absolutely need the myelin for quick conduction of the action potential. One of my best friends that I grew up with, she has a little boy, Otto. Otto is, oh gosh, he just turned 8. When he was 2, he got this acute condition called transverse myelitis. Has anyone here heard of transverse myelitis? It's really rare. And it's an autoimmune disorder, right? So his own immune system attacked a transverse section of basically the myelinating cells in his spinal cord. And so what happened is it demyelinated a bunch of cells in his lower spine, and he basically, over the span of a few days, went from a kid who was running around to paralyzed from the waist down. And so it's totally crazy condition. They still don't really have a handle on what causes it. 30% of people apparently fully recover. I had a student in my class, when the first time they told this story, was like, oh yeah, I had transverse myelitis, but she fully recovered from it. Some people partially recover, and then 30% don't recover at all. So Otto, unfortunately, was one of the unlucky 30% who didn't recover. He's still, he's an amazing, creative, exuberant kid, but it is just shitty, right? Okay. It's the right word for that. It's shitty. I wasn't going to say poopy because it's shitty. All right. And so when the action potential fires, what ends up happening is that synaptic vesicles that have a bunch of neurotransmitters ready and waiting, they end up fusing with the axon terminal and, right, traversing the synapse and being received by the postsynaptic cell. So a lot of times when you talk to people who are referring to neurons, they'll refer to the presynaptic cell, postsynaptic cell. Presynaptic cell, it's on the side of the synapse that's letting go of the neurotransmitter. Postsynaptic cell, right, other side of the synapse, receiving the neurotransmitter. All right, and with that, we can move on to lecture 6-7. Okay, how'd the learning module and quiz go for you guys? You guys appreciating having 2 chances on the quizzes?
Yes.
I had a student who came to me